So good morning to all. So in the last class we discussed about the DNA, different properties of the DNA and how to calculate the length of the DNA and how to calculate number of base pairs and how to calculate the percentage of different nitrogen bases in the DNA, in the given DNA, and how to calculate the number of phosphodiester bonds. Okay. So next what we discussed in here, double strand DNA. So discovery already discussed that. So the double helical structure of DNA explained by the Watson-Crick. So double helical structure of DNA explained by. So Watson-Crick based on the X-ray diffraction data. That is X-ray diffraction data produced by so Wilkins and Franklin. So they are the two scientists. They given that X-ray diffraction data. So based on that X-ray diffraction data, Watson-Crick explained secondary helical structure of DNA. This secondary helical structure of DNA rotates, or it is a right-hand helix. So it is a right-hand helix. So see in the picture, it is rotated in the right-hand side. So it is a right-hand helix. Got it? So according to them here, the DNA is of two polynucleotide chains. So DNA is of two polynucleotide chains. It is made up of nucleotides. So nucleotides consists of three components. There are three components which are present in the nucleotides. What are the three components? We have to start always from the nitrogen bases, then followed by pentose sugar, then followed by the phosphate. Okay. So these two strands are anti-parallel each other. Okay, not parallel. So what is a complementary base pairing is present. So what is the principle behind the transcription? What is the principle behind the replication or duplication of DNA? That is nothing but complementarity. So remember, so complementarity is the principle behind the replication, as well as complementarity is the principle behind the transcription also. A plates with thymine, 1 plates with cytosine. Got it? So what is that? So here as well as what is the main hallmark of this Watson-Crick hypothesis? That is nothing but base pair. So he given that base pairing. So based on the this one of the law, that is the Chargaff rule. Based on that Chargaff rule, so they given that base pairing process, base pairing proportion. So here, what is that Chargaff rule? So here, according to the Chargaff. So according to the Chargaff, so number of. So what is the Chargaff rule here? So according to the Erwin Chargaff, so the ratio of ratio of adenine and thymine. Remember the ratio of adenine and thymine and ratio of guanine and cytosine. Always constant. Always constant. So here remember the ratio of adenine and thymine and ratio of guanine and cytosine always constant and equal to 1. Okay, so this is Erwin Chargaff rule. So based on this, it is possible to calculate also. For example, in a given DNA, adenine percentage is 20, automatic thymine percentage is 20. So the remaining 60%, the remaining is the 60%, 30% goes to guanine and 30% goes to cytosine. Okay, this is Erwin Chargaff rule. Okay, so next, who found that the presence of a acidic substance inside the nucleus? So inside the nucleus, what is present? One of the acidic substance is present. So that he called that nuclein. Okay, so Friedrich Miescher. So Friedrich Miescher, he found that presence of acidic substance inside the nucleus. That we already discussed that. That's why I am skipping here. And the next one here. So the equal distance present between the two polynucleotide chains. So equal distance present between the two polynucleotide chains. So the equal distance due to the presence of hydrogen bonds. So equal distance due to the presence of hydrogen bonds. So hydrogen bonds are also responsible for the stability of DNA. So they are appear like stacks. For example, if any given DNA consists of high guanine cytosine ratio, more hydrogen bonds are present. If any in the given DNA, the DNA consists of, if the DNA consists of high guanine cytosine ratio, that DNA consists of more hydrogen bonds. And that DNA resistant to heat also. I remember in polymerase chain reaction, we are exposing the DNA to the heating process at 94 degrees. What will happen? The two strands are separated from each other. Okay. If DNA consists of more hydrogen bonds required more energy or more heat to separate two DNA strands. If the given DNA consists of high number of guanine cytosine, because of high number of hydrogen bonds, more number of hydrogen bonds, and resistance to heat. So equal distance due to the presence of hydrogen bonds and stability of DNA due to the hydrogen bonds. Next. So dozen types of DNAs which are present on our body. So double hand helix, okay, right hand helix. That BDNA especially. BDNA is the right hand helix. And this is a secondary helical structure of DNA. So what we are seeing, that is a secondary helical structure because it consists of two polynucleotide chains. All the DNAs, like, so here all DNAs, dozen type of DNAs are present in our body. So they are classified into different types based on number of hydrogen bonds present per helix. So they are classified based on number of hydrogen bonds present per helix. For example, here ddNA consists of 8. Sorry. So here the DNA classified different types based on the presence of number of base pair per turn. Number of base pairs per turn here. So ddNA consists of 8 bases. ddNA consists of 8 bases. cDNA 9 bases. bDNA 10 base pairs. And aDNA consists of 11 base pairs. zDNA consists of 12 base pairs. And here all the DNAs here, they are the right hand helix. They form the right hand helix except zDNA. zDNA only, it is a left handed helix. So zDNA in the form of zigzag manner. So zDNA in the form of zigzag. Got it? So what is the here ascending order of DNA here? So this is ascending order. So here ddNA consists of 8 base pair per turn. So what is the length of that complete pitch? That is a 3.4 nanometer. If you convert that 3.4 nanometer into angstrom, it is become 34 angstrom. So the distance between the two base pairs here, this is one base pair, this is one base pair. So what is that? The distance, the length of this one, that is 0.34 nanometer. So based on this, it is possible to calculate the length. So our DNA, so diploid cell consists of 6.6 into 10 to the power of 9 base pairs. If you convert that 6.6 into 10 to the power of 9 base pair into meters, that is become 2.2 meters. Same E. coli also 4.6 into 10 to the power of 6 base pairs. If you convert that, it become millimeters. 1.32 millimeters. That is the length of that E. coli chain. So all this type of DNA is present in our cells as well as all the cells also. So this classification based on number of base pair per helix or complete pitch. And here ZDNA consists of high number of base pairs, that is 12 base pairs, and it is a left hand helix. It is not right hand helix, it is a left hand helix. That is only one left hand helix, and it is in the form of zigzag also. That's why it is called as ZDNA. So one more important concept, that is central dogma. So here central dogma concept is given by Francis Crick. What is that generally central dogma? So we already discussed that DNA is the best for the storage of genetic material because it is more stable, less reactive. For storage of genetic information, DNA is best than compared to the RNA. For flow of genetic information, for expression of genetic information, RNA is better than the DNA. DNA depending on the RNA to express its character or to flow the genetic information. For example, from the DNA, RNA synthesized, RNA responsible for the synthesis of proteins. So the flow of genetic information from DNA to RNA to proteins, we call as central dogma. And one more important character of DNA as well as RNA. So they undergo self-replication inside the nucleus. They have capability to undergo replication. The viruses also, RNA has capability to undergo replication. Got it? This one, flow of genetic information from DNA to RNA. So see here, the synthesis of RNA from DNA we called as transcription. The RNA always in the eukaryotes, that is single strand, not double strand. So synthesis of proteins that we called as translation. So we get that central dogma, that flow of genetic information from DNA to RNA, RNA to proteins. Maximum all the living cells, this is a flow of genetic information. But except some viruses, in some viruses, the RNA is a genetic material. In such cases, the flow of genetic information from RNA to DNA. So now what we are seeing in this video, that is flow of genetic information from DNA to RNA, that is transcription. Next, RNA to proteins, or we call that translation process. So next process here, reverse transcription. So as I told you, in the viruses, the flow of genetic information from RNA to the flow of genetic information, that is from RNA to RNA to DNA. In some viruses, the flow of genetic information, that is from RNA to DNA. So synthesis of DNA from RNA we call as reverse transcription. We are simply called as RT. So this is very important bit. Synthesis of DNA from RNA we call that reverse transcription. So this DNA, also known as the DNA that is synthesized from RNA, also known as cDNA, complementary DNA. So maximum in the examination they will give like that cDNA. So whatever that DNA is synthesized from RNA through the reverse transcription we call as cDNA, complementary DNA. Okay. So here the enzyme required for synthesis of DNA from RNA, nothing but reverse transcriptation. So what is the enzyme required here? That is nothing but reverse transcriptation. What is the enzyme required for synthesis of DNA from RNA? Reverse transcription. So here the flow of genetic information from RNA to DNA reverse transcription only in some viruses, not all viruses. Those viruses they contain RNA is a genome, genetic material. So what is the other name for here reverse transcriptase? What is the other name for reverse transcriptase? So this is RNA dependent. So here RNA dependent DNA polymerase enzyme. What is the enzyme other name for reverse transcriptase? RNA dependent DNA polymerase. Why it is called as like that RNA dependent? It is depending on the RNA for the synthesis of DNA. So that's why it is called as RNA dependent DNA polymerase enzyme. So reverse transcription, synthesis of DNA, the DNA cDNA from RNA is called as reverse transcription. So RNA always single strand. So what we are seeing that round shape one reverse transcriptase enzyme and resulting synthesize DNA, single strand DNA again become double strand DNA. That is not required for you, had to forget. So just you remember that synthesis of DNA from RNA. So this is very important for reverse transcriptase enzyme required for reverse transcription. Generally, in all the cells, the flow of genetic information from DNA to RNA, RNA to proteins. But here, RNA to DNA, DNA to RNA, again proteins. So this one packaging of DNA helix. So in the eukaryotic cell, so what is the size of the nucleus in the prokaryotic cells? So this is the size of the nucleus, the normal size of the nucleus in the prokaryotes. So that is around 10 to the power of -6 meters. This is the size of the nucleus in the eukaryotes. So this is eukaryotic. So eukaryotic cell nucleus size. So nucleus size. Okay. So here, especially the human beings, imagine our human beings, the nucleus is size is 10 to the power of -6 meters. Then what is the length of the DNA in the diploid cells? That is around 2.2 meters. So here 2.2 meters, how it is possible to pack inside the nucleus? Because the nucleus size is very too small. So even though it is the DNA, our DNA tightly packed inside the nucleus. Okay. So for packing of DNA, different types of histone proteins. So for packing of DNA, different types of histone proteins which are involved, not only histone proteins, some non-histone chromosomal proteins also involved in higher level of packing of DNA. So both histone proteins as well as non-histone proteins which are involved in the packaging of DNA in the eukaryotic nucleus. So NHC proteins, that is nothing but non-histone chromosomal proteins, they required for higher level of packing. So this is very important, higher level of packing of, so higher level of packaging of DNA. So what are the required for the higher level of packaging of DNA? Non-histone chromosomal proteins required. So in the E. coli also, now what you are seeing this picture, this one is E. coli cell. So in the E. coli cell, only one chromosome is present. Or generally in the prokaryotes, only one chromosome is present. So single chromosome is present. So that single chromosomes that is in the form of organized in the form of a large loops. Only single chromosome is present. Along with single chromosomes, plasmids also is present. Extra chromosomal molecules. So we already discussed in the recombinant DNA technology. We are using these plasmids as a vector. So this single chromosome is naked one without any histone proteins. So naked. Because this chromosome without any histone proteins. Yeah. So especially in the bacteria cell, all bacteria cells, only one chromosome is present. That is double stranded DNA. Double stranded DNA that is in the form of circular without histone proteins. That's why it is called as naked one. So how it is in the form of circular. This is double strand DNA. So double strand DNA. Okay. So this is organized in the form of large loops. So it is not scattered in the cytoplasm. It is not scattered throughout the cytoplasm. It is held at one place. It's not scattered in the cytoplasm. It is held at one place. That place, that region we call it as nucleoid. What is that nucleoid? Where that chromosome is located, that region, the place in the bacterial cell cytoplasm, we call as nucleoid. So it is held at one place. It is held at one place. It is held at one place by the involvement of non-histone proteins or positively charged proteins. Positively charged proteins. It is held at one place by positively charged proteins. It is not scattered in the cytoplasm. It is held at one place. That place we call as nucleoid. So now we are discussing about that prokaryotic genome that is present freely in the cytoplasm but is not scattered. It is held at one place by the positively charged proteins. So along this one, so we already idea plasmids also is present. So double strand DNA extra chromosomal molecules replicate independently to the chromosome. So these are not histone proteins, they are the positively charged proteins, similar to like that of histone proteins. Histone-like proteins are present. So one more important character here, nucleosomes absent. Nucleosomes absent. So nucleosomes only present in eukaryotes. So nucleosomes absent here. So nucleosomes which are involved in the packing of DNA in the eukaryotic cell. So not only nucleosomes are made up of histone proteins. Okay. So next here, packing of DNA in the eukaryotes. So in the eukaryotes, a special type of proteins involved. That is nothing but histone proteins. These are the positively charged proteins. Histone proteins are the positively charged proteins. How many different types of histone proteins are present? That is histone protein 1 and histone protein 2A and histone protein 2B and 3 and 4. So total 5 different types of histone proteins are present. So this consists of all different types of amino acid, but it consists of more abundant amino acid, basic amino acids like lysine and arginine. So these proteins consists of high percentage of basic amino acids. High percentage of basic amino acids. So these amino acid side chains they carries positive charge. Where the positive charge are present on these amino acids? Side chains. Side chains of this amino acid they carry positive charge. That's why due to the presence of high number of this one lysine and arginine, so that histone proteins are we call as basic proteins. They carry the positive charge. So histone proteins are present in the eukaryotes. Histone-like proteins are present. So I told you here nucleotide held at one place by the involvement of positively charged proteins. They are similar to like that of histone proteins, but not exactly histone proteins. So here all these histone proteins, especially here. So two copies of this one, all these histone proteins, that is H2A, H2B, and 3 and 4. So which are two copies of each histone proteins. So two copies of each histone proteins which are involved in the formation of octamer. This is octamer. So the other side, back side. So this one back side. So imagine this one is H2A. This one also H2A. This one H2B. This one also H2B. And next here H3, H3, H4, H4. Okay. So all these histone proteins which are involved in the formation of octamer. So this is octamer. Octamer consists of 8 histone proteins. Okay. So around the octamer, the DNA is coiled how many times? 2 times. The DNA is coiled how many times? 2 times. So octamer plus DNA, now what we are calling that? Octamer plus DNA we are called as nucleosome. So this is very important. So in the last KSET examination, what they given? So octamer is present. Around the octamer, the DNA is coiled how many times? 2 times. Okay. So what is the length of DNA that is coiled around the octamer? That is nothing but 200 base pair length. Around the octamer, what is the length of DNA? The DNA is coiled around the octamer. That is 200 base pairs. So this is now DNA plus octamer. It is called as nucleosome. So one more nucleosome. Okay. So here H1 protein. So here this one is H1. Histone protein 1. Okay. This is one more nucleosome. This is H1. So imagine this is one nucleosome and this is H1. So one more nucleosome. Okay. So this is H1. So between the two nucleosomes, DNA is present. Between the two nucleosomes, DNA is present. So this DNA is called as linker DNA. Very, very important. The NCERT is not given. What is the DNA is present between these two nucleosomes? Linker DNA is present. How many times DNA is coiled around the nucleosome? Sorry, octamer, 2 times. What is the length of the DNA that is coiled around the octamer? That is 200 base pair length. So recently in the case it is given this 200 base pairs. Got it. So next one, what is solenoid? Okay. 6 nucleosomes together they form a solenoid. So I already told you nucleosome is made up of octamer plus DNA. How many histone proteins are present in the octamer? 8 histone proteins. Actually 4 histone proteins. 2 copies of every histone proteins they form a octamer. This octamer consists of 4 different types of histone proteins. 4 different types of histone proteins. That we have the idea. Okay. Total, how many histone proteins? 8 histone proteins are present. So all nucleosomes. So 6 nucleosomes they form the solenoid. We have the idea. Leave it that one, but you have to remember. But what I am here, what I am telling here, all nucleosomes together they form. How it is appear? It is appear like a beads on string. So it is appear like a beads on string. So how the nucleosomes on DNA appear? They appear like a beads on string under electron microscope. Remember this one, not under normal microscope, under electron. So under electron microscope, they appear like a beads on string. So remember, this is very important, not under normal microscope, under electron microscope, they appear like a beads on string. So between the two, this is nucleosome, this is nucleosome. What is the DNA is present? Linker DNA is present. This is a linker DNA. So all nucleosomes together they form a chromatin thread. Chromatin thread. So all nucleosomes together they form a chromatin thread. But you have to remember solenoid consists of 6 nucleosomes. Remember that one. Just okay. Leave it that one. So here what I am discussing here, all nucleosomes together they form a chromatin thread. Or all nucleosomes condensed they form a chromatin thread. Got it? So here chromatin thread. So during the mitosis, what will happen? Prophase, the chromatin fiber condense and form the chromosome that you have to remember. Interphase DNA in the form of chromatin fiber. So what I am telling here, all nucleosomes together they form a chromatin thread. Chromatin thread then followed by chromatin fiber. Chromatin fiber. chromatin thread, then followed by chromatin fiber. So the DNA in the interphase, remember that cell cycle, how many phases are present? Interphase and mitosis. Again, interphase classified into three phases. One is gap 1, S phase, and G2 phase. So cell cycle is very important. Protein synthesis takes place in the gap 2. DNA synthesis takes place in the S phase. In the gap 1, organelles become double in G1 phase. Cell growth through the interphase. I will discuss later clearly about next one. So first we have to remember chromatin thread followed by chromatin fiber. How the nucleosomes along with DNA uses appear in electron microscope. Beads on string. Yeah. So next one. Yeah. So first, so DNA in the form of, in the interface, DNA in the form of chromatin fibers. Okay. So here chromatin fibers, when the cell enter into the mitosis, especially in the prophase, the chromatin fibers start condensing. Condensation of chromatin fibers completed in the metaphase, early metaphase. You have to read regularly about this one, otherwise you are going to be forget. So chromatin fibers are directly writing chromatin fibers condensed and form chromosomes during mitosis. Especially so during mitosis as well as meiosis also. In the meiosis prophase also chromatin fibers condense and form the chromosomes. Okay, and form the chromosomes when especially in the mitosis. So here condensation completed in early metaphase. So condensation of chromosomes completed in early metaphase. So first we have to start with here nucleotide. All nucleotides they form polynucleotide chains. The two polynucleotide chains they form the DNA. So DNA, next DNA plus octamer form nucleosome. So all nucleosomes they form, I'm directly writing chromatin thread. So they form a chromatin thread. So that chromatin thread again form chromatin fiber. Chromatin fibers again form the condense and form chromosome. So all chromosomes together they form genome. So this is the order. So important things, histone proteins. So they also have abundant lysine and arginine. Side chains they carry the positive charge, not amino acids. So actually here amino acid consists of amino group or carboxyl group. So they are attached to the alpha carbon. That's why amino acid, what we are calling that alpha amino acids. Remember that carboxyl group, amino group, they are attached to the first carbon or alpha carbon. That's why all amino acids, 20 different amino acids are alpha amino acids. So here the variable group is present, that is R. The variable group may be hydroxyl group or methyl hydroxymethyl. Okay. So based on that, 20 different amino acids. So next, higher level of packing. So up to here, what we discussed, how the DNA is coiled around the octamer, and then followed by nucleosomes, and then followed by chromatin fiber. Chromatin fiber condensed form chromosomes. Okay, when the condensation occurs, especially in the mitosis and meiosis, especially the condensation over at condensation of chromatin fiber starts in the prophase, completed in early metaphase. Remember, condensation completed in the early metaphase. Okay. So higher level of packing required. Yes, higher level of packing required. Non-histone chromosomal proteins. Remember, packaging of DNA required histone proteins. Higher level required non-histone chromosomal proteins. Okay. So next here. So nucleosome. So this is given. Draw the structure of nucleosome. So how many times here DNA is coiled? 2 times. There is H1 histone protein present outside of octamer. So histone protein present outside the octamer. So this is octamer. DNA coiled around 2 times. Then what is the length? Average length of this DNA? 200 base pairs. Then how many nucleosomes are present? How to calculate how many nucleosomes? How to calculate how many nucleosomes are present in the given DNA? So then how it is possible to calculate? For example, human genome consists of how many nucleosomes? Okay, just we have to 6.6 into 10 to the power of 9 base pairs. Yeah, 200 base pairs. When you divided this number of base pairs with 200 base pairs, you will get some value. So around 0.33 into 10 to the power of 9 nucleosomes are present. How many nucleosomes present in E. coli? How many nucleosomes present in E. coli? No nucleosomes. Nucleosomes are absent. Nucleosomes, no nucleosomes are present. Remember, in prokaryotes, how many nucleosomes means? There is 10 nucleosomes are present. And how to get number of nucleosomes are present per cell? For example, examination they will be like that. If the length of the DNA is 3.6 into 10 to the power of 9 base pairs, then what is the how many number of nucleosomes are present? This is the length of DNA of one any given organism. Then how many nucleosomes are present? Just you have to divide it this number of base pairs with 200 base pairs. You will get some value. So our cells consists of how many nucleosomes? 0.33 into 10 to the power of 9 nucleosomes are present. So one more important. So this you have the idea. Total 8 histone proteins, 4 types of histone proteins which are involved in the formation of octamer. DNA plus octamer we call as nucleosome. Yeah. So next one is euchromatin and heterochromatin. So this is very important. What is that euchromatin and heterochromatin? So tightly packed DNA or densely packed DNA we call as heterochromatin. So loosely packed DNA we call as euchromatin. So if DNA is tightly packed, that is transcriptionally inactive. So transcriptionally inactive. Which one is transcriptionally inactive? So here heterochromatin is transcriptionally inactive. So due to the packing, it stains dark. It stains dark. For example, acetocarmine we are using for the staining of the DNA. So what is the dye which are used for the staining of DNA? Acetocarmine. Acetocarmine also known as acetoarsine. So this is a basic dye. This is basic dye. That's why it is going to be bind to DNA. Acetocarmine used for the staining of the DNA. So several stains are there. In our laboratory, what we are using to locate the nucleus, to locate the chromosome, that is nothing but acetocarmine. So when you apply acetocarmine, the nucleus or chromatin fibers appears red or pinky color. Red or pink color. So here heterochromatin is transcriptionally inactive. It is tightly packed. It is tightly packed DNA and stains dark. So exactly euchromatin is transcriptionally active one. So loosely packed one and it stains light. So these are the functions of euchromatin and heterochromatin. So what are the features means which one is transcriptionally active? Euchromatin. Why it is transcriptionally active? Because it is loosely packed. So easily undergo. So that's why easily undergo transcription process and can synthesize RNA. If it is tightly packed, not possible to synthesize mRNA. That's why it is transcriptionally inactive. So which of the dye used for staining of chromosomes or chromatin fibers? Acetocarmine. It is a basic dye. So heterochromatin stains very dark and euchromatin stains very light because it is loosely packed. Next, transforming principle. Yeah. Still up to 1929, they don't know exact idea what is exactly genetic material, whether DNA, proteins, or RNA. Okay, so transforming principle. So George Mendel, he worked on. So we have the idea. So he went the principles, inheritance principle, law of dominance, law of segregation. So Walter Sutton also, Theodore Boveri, remember that Walter Sutton, Theodore Boveri, these argued that and they noted that the behavior of the genes parallel to the behavior of chromosome or behavior of chromosome parallel to the behavior of genes. T.H. Morgan, you got idea T.H. Morgan? Linkage and recombination. He worked on one of the fly that is fruit fly, that is Drosophila melanogaster, who proved that chromosomal theory of inheritance. T.H. Morgan. Experimental proof is given by Thomas T.H. Morgan. Remember this one, very important. Okay. So when they are doing experiments on different areas, so they found that genetic material that is located inside the nucleus or chromosomes, they are located in the inside the nucleus of the most cells, but they don't know exact what is the genetic material, whether DNA, RNA, or proteins. Okay. So that the proof, the first time one of the person he attempt some experiments by using one of the bacteria, Streptococcus pneumonia bacteria. The scientist nothing but Frederick Griffith. So he performed experiments, especially in 1928, on one of the bacteria that is Streptococcus pneumonia. So it caused pneumonia infection. Streptococcus pneumonia caused a lung infection in the animals as well as human beings also. Streptococcus pneumonia, it caused lung infection. So it is a cocci bacteria. So this one Streptococcus is a cocci. Then why it is called as Streptococcus? So it is appear like a long chain. That's why it is called as Streptococcus. So it is a cocci. Cocci means the bacteria round shape. So this bacteria appear like a chain. That's why it is called as Streptococcus. It's called as Streptococcus, cocci bacteria. So this bacteria also known as Diplococcus. What is the other name for this bacteria? Diplococcus pneumonia. So now we are calling as Streptococcus pneumonia. So Griffith worked on one of the disease that is pneumonia. Griffith found vaccine against one disease that is pneumonia. And he found that that caused is nothing but Streptococcus pneumonia. It caused lung infection in the all animals as well as humans. So there are two types of strains which are present in Streptococcus pneumonia. There are two strains which are present in the Streptococcus pneumonia. So what is the two? So there are two strains which are present in Streptococcus pneumonia. One strain is here, R strain, next one is S strain. So there are two strains which are present in Streptococcus pneumonia. One is that you have the idea, this one. So one is R strain and S strain. So they are the two different strains which are present in Streptococcus pneumonia. It causes pneumonia infection. So Frederick worked on one of the disease pneumonia. He found a vaccine against one of the disease that is pneumonia. And to prove that transformation, he used one bacteria that is Streptococcus pneumonia. This is not virus, this is bacteria. What is the other name for this bacteria? Diplococcus. So what is the two strains here? So which one is virulent? which one is non-virulent. Here what we are seeing S or stain is avirulent or non-virulent, means it does not cause any pneumonia infection. That's why it is avirulent or non-pathogenic also. So here R stain cause never any infection. Here S stain cause pneumonia. That's why it is called as virulent. So here S stain is virulent, R stain is non-virulent. So here R stain cause produce a rough colonies. So these are the rough colonies. When these bacteria, when they grow on agar media, that is nutrient agar media. So imagine this is a petri plate or a plate, glass plate, petri dish. It consists of media, growth media. This growth media, all stain produces rough colonies. These are the rough colonies. S stain produces smooth colonies. So why here S stain produces smooth color colonies? Are you observing here? It consists of one of the coat, mucus coat. So S stain consists of mucus coat. That's why it is producing smooth colonies. So mucus coat is present. So mucus coat is made up of polysaccharides. What is that polysaccharide? So that is nothing but carbohydrate. Homopolysaccharide. Sorry, not homopolysaccharide. This is heteropolysaccharide. Okay. So tomorrow we will discuss the remaining part. If you have any doubts, please ask. Yes, anyone? Okay, thank you.